Hello, in this video we will discuss about the PCR, polymerase chain reaction, and what is the significance of the PCR. So basically the PCR generates copy of DNA. So the PCR can be classified the different type of PCR we will discuss in this video. But important to think about that is the reverse transcriptional PCR. This reverse transcriptional PCR used for the detection of the viral infection through RNA. So it is also used for the RNA to convert DNA, that is the complementary DNA, will use for the amplification of that genes, which that is the active in the specific cell. So here is the quantitative reverse transcriptional PCR, and it is the quantitative, show the quantity, and here is the reverse transcriptional quantitative PCR, and that is the another is the quantitative PCR. Let's begin to understand with the step and procedure of reverse transcriptase PCR. So important thing is that the show the quantity of the DNA amplification is known as the quantitative. So the qualitative is the meaning of the quality, but quantitative meaning the quantity, the number of the segment will amplify. So here is the sample preparation in the first step and selection of primers. Selection of primer means our desired primer, which that will be bound with a specific region of the for example, messenger RNA, if you are using the messenger RNA or RNA. So on the other hand, here is the reaction preparation, a real-time PCR, cyclic condition, and strand synthesis is the all basic requirement for the amplification of the gene. So let's begin to understand the selection of primers can be random, oligo, deoxynucleotide, and the sequence specific. So a sequence specific can be the specific gene will be amplified and other gene will not amplify. It means a specific. And random mean it is the segment of the RNA, for example, which that will be bound with a randomly. So in this way the gene will be amplified with a randomly because, for example, we are using the primer for amplification of the certain genes. So the random primer will be bound to amplify that genes. While in the case of the, here is the oligo and random is same. So the sequence specific primer can be gene specific. For example, we are amplifying the insulin producing gene. So the primer should be related to the insulin producing gene and where the binding with the specific gene for amplification of that gene. So that is why the amplification of, for example, insulin producing gene will be amplified, and we can check that and we can comparison. And on the other hand, the biotechnological field which that is used for these things. So here is the principle of reverse transcriptional PCR we will discuss. Reverse transcriptional PCR means the reverse transcription of the RNA into the DNA. And the reverse transcription enzyme synthesizes the complementary DNA from the RNA. The process has two steps, the reverse transcription progress, and the second multiplication and quantification will be occur. So the multiplication and quantity will be shown. And in this way here you can see that is the all things we are understanding in this diagram to understand. This is the EP tube or some tube or any tube, test tube for containing a reverse transcriptase enzyme addition, messenger RNA, for example the insulin producing messenger RNA, or infected cell containing the RNA, which that is for example coronavirus. So that we are understanding also the coronavirus infection detection. So the buffer here, buffer we can use, and here is the deoxynucleotide triphosphate, which that is also used. So in this way here is the structure of the deoxynucleotide will be used, and sequence specific primer we can use. Also the DNA polymerase. DNA polymerase means the DNA will be polymerase enzyme used for it. And in this way the one step real reverse transcriptase PCR, reverse transcription PCR will be used. And in this way here is the manual PCR machine discovered in invention in 1985, and it is the contain a denaturation step 95-degree required, annealing process 60-degree, and the extension required the 72-degree Celsius temperature. For example, the step 2 means the two-step stages is the another, and here the thermal cycler automated PCR invented in 1988. So here is the two-step PCR we can also use. In this way, here is the further which that is using the DNA polymerase and buffer, and on the other hand the deoxynucleotide triphosphate and sequence specific primer we can use. In this case, the step 1 is not used, a sequence specific also used, but in this case here is the again which that is used, that is we are not adding the messenger RNA, but we are DNA polymerase buffer, and again that is we can complementary DNA produce. So this is the thermal cycle, the quantification of complementary DNA, and we can add the tag polymerase. So in this way the quantification of complementary DNA and tag polymerase is used for it. So let's begin to understand the manual PCR or some thermal cycler function. So here is we are extracting the infected cells RNA or messenger RNA, isolation of total RNA or messenger RNA. After the isolation, so here is the poly A tail will bind with the primer. The TTT is the rich content of the primer. After the binding with the rich is the also the G guanine, cytosine, and adenine are present, but is the beginning to RNA to become a DNA, the complementary DNA. It is known as the complementary DNA because it is again will be transcribed into the messenger RNA. That messenger RNA will be equal to our desired RNA. So let's begin to understand the first cycle. The PCR will amplify by PCR to produce the copies of the DNA segment. This copy of the DNA segment is the complementary DNA. After this, the extension, after the denaturation will be extension. So this is the PCR process. And previous process is not PCR, but it can be synthesis the complementary DNA. So in this way, here the primer will be bind and polymerase enzyme, tag polymerase used for the extension after the annealing. So the annealing means the primer will be bind. So it will be amplify more and more genes. So that is why the amplification of the genes, the polymerase chain reaction will be occur. So this reaction will amplify more and more gene segment. So let's begin to understand the two type of polymerase chain reaction. So the fluorescent dye-based reverse transcriptase dye-based real-time PCR, and the DNA probe-based real-time PCR. So the reverse transcription, it also after the reverse transcription for detection of the specific gene. So here is the DNA which contain deoxy double-stranded DNA template DNA, and this denaturation will be occur. After the denaturation, we are adding a ground state in fluorophore. This ground state fluorophore will be bind when with the DNA, so the excited state fluorophore will be occur and produce the color. So after this denaturation, to separate two single-stranded DNA, and in this way the fluorophore will be bind. And fluorophore bind when the DNA will be formed. Remember about that. After the annealing process, the primer anneal with the 5' direction to 3' direction of the DNA anti-parallel. And in this way, you can see that is after the complete synthesis of the double-stranded DNA will be bind with the fluorophore. So the quantity of the fluorophore we are adding with our measurement, and in this way the primer extension to produce color after the binding with the DNA. So that color will be more increase when the increase of the color through light. So the increase of the color will recognize and identify us about the quantity of the DNA segment amplifications or gene amplification, and we can check through color if the color production more. So that is why the primer extension and the gene amplification with the specific gene amplification will be more. So the DNA probe based real-time PCR is the separate things. In this way, here is the double-stranded DNA template, which that is our desire for amplification. And here is the light and the DNA probe which we can use, which contain cancer and fluorophore. In this way, the cancer will be absorb the light and transmit the light to the cancer, but it will not emit light, so it will colorless. But it will be separate, so the cancer will be separate to fluorophore produce color. So here is the denaturation of the DNA again to annealing process, or during the annealing process, so the cancer containing DNA probe will be bind during that single stranded DNA. After this binding with a specific region and after the complete amplification and annealing to extension process to become a tag polymerase enzyme used for the cutting of this cancer, and after this cutting of this cancer to release the fluorophore, and after this fluorophore we can be bound with the DNA or is released to produce color. So and color will be more production of the color, so the cancer will be specific color containing our desired. So the color when produced more, that type of color will show that this gene is amplifying. But in this case the color will produce more, so the quantity will be more, and color will produce less, so the quantity will be less of the polymerase chain reaction to amplify the segment. So here is the viral infection, for example the coronavirus. So the virus sample we take and RNA extraction, RNA extension will be occur after the extraction, and in this way the same mechanism, the reverse transcription to become a DNA. That DNA will be complementary DNA. After here is the cycle, horizontally show that diagram, and in this graph, and here is the fluorescence. Fluorescence quantity will be show. The more fluorescence show, that means the infection chance is positive. So here is the threshold line will cross, so the CQ value will be occur. And in this way here is the more amplifying and fluorescence will be produced more, and the cycle will when more. So the more cycle will produce more in fluorescence. In this way, you can think about that. This gene, that means if we will provide the specific primer for amplifying the specific gene, so the coronavirus gene, if present, so the amplifying more segment, but in this case if not, so the not more quantification of the color. So here is the non-exponential growth will be occur of this graph, and in this way the exponential phase will show us that amplifying the gene which that related to the disease causing agent are present in the infected person. So that is why the non-exponential growth means the equal color will be show, and that means the maintain and not more color producing system. So after this here is we are taking the sample and checking the coronavirus disease. So here is the getting the sample and extraction of RNA. After the extraction of RNA, amplification of RNA and positive or negative detection through result. And here we can introducing the probe is the orange color. So here you can see the negative test is the white color, while the positive test will show the orange color. So let's begin to draw a graph again for days 1 to day 16 for understanding during the infection, the pre-infection, after, during infection and post-infection time. So here I am drawing the line, upper line show that the low analytical sensitivity PCR through PCR and high analytical sensitivity through PCR in the down line. So here is the pre-infection positive by PCR will show that is the color, and in this way is you can see that is basically exponential growth of the polymerase chain reaction will amplify the segment. It means the positive because the segment is present, which that is our desire for the amplification. So the color will produce more through probes. So in this way here is the viral load will be more. In this way the viral load will be more, and it means the here is the days and the no infection here, and it means the graph will not show the color, while here the viral load more, so it means the peak of the graph will be more. so that is during the infection time the viral load will be more. After the recovery time, the viral load will be decreased and the positive infectious positive by PCR can be detected after the infection time. So it can be the polymerase chain reaction is also used for the post-infection time and pre-infection time as well as the during infection time for checking the more growth of this graph for understanding. So that is why it is the probe is very important for the checking of the viral infection due to the amplification of that gene. So that gene will be amplified more. It means we can think about that gene are present in the infected cell. So thanks for watching. Please make sure to subscribe, like and share. Ask the question for answer and suggest me to another video.